I think with synthetic biology and all the AI tools and all these protein engineering
Mm-hmm.
progress that we have seen recently, also in the Nobel Prize thing,
Yeah.
I think the materials can have the decision making ability,
Oh,
so they know when to interact, with what to interact, and where to interact.
okay. Hello, everyone. Welcome to the Material Minds podcast, the place where we talk to the people exploring new materials for a better world. Today, we talk with Professor Luai Khoury from Technion, the Israel Institute of Technology. We talk about his research in developing protein based materials. He creates sponges with catalytic activity, protein sponges with catalytic activity. He talks about making soft robots that can deliver drugs inside your body, inside your gut, and also about an espresso inspired machine for making protein microgels. It was a very curious and bio inspired conversation that we had, and I'm sure you're going to enjoy it too. So let's go ahead. Hi, Luai. Welcome to Material Minds.
Hi, Shrish. Thank you for having me today. It's a real pleasure to be a dynamic and share with you what we do in the material science and engineering department at the Technion.
Yeah, I'm really curious to hear what about your background, what you do, and so on. And of course, we're going to start with our favorite game, Three Truths and a Lie.
Hmm.
So you're going to tell me four statements. You prepared four statements.
Yeah.
You're going to tell me four statements, and I have to figure out which one is the lie. Three are going to
Okay.
be true, one's a lie. I have to figure it out. I'm going to ask you some questions in the middle, like after every statement,
Uh-huh.
to try to poke into find out the hidden meanings behind it or so on. And so yeah, if you're ready, I'm ready for the
All
first
right.
statement.
All right. Let's play the three throws and the light game.
Hmm.
And I will try to be smart enough, like my materials,
Yeah.
to fool you or
Hmm.
to make it difficult for you to find the truth or the lie. So the first statement is I once created a protein based material
Hmm.
or sponge
Hmm.
that makes or keeps trypsin active
Hmm.
for over 10 days.
Hmm.
And I can use it for like 40 times for its application.
Okay, so you have a sponge. When you say sponge, and like in terms of your material, your protein-based materials,
Mm-hmm.
when you say sponge, is this like a kitchen sponge that I can think of?
Yeah,
Okay, it's going to be like a kitchen sponge. And you're saying trypsin is active. Is the trypsin within the sponge?
uh huh, right.
Okay, the trypsin's within the sponge. Um, what does the trypsin do?
Trypsin is an enzyme, digesting enzyme that cleaves the proteins into protein fragments.
Okay, so trypsin basically cleaves other proteins, and so your application is probably to take it and cleave other proteins in some location. Is
Mm-hmm.
that the case?
Yeah,
Okay.
almost.
Would you be willing to share what kind of proteins you want to cut with it with the sponge?
We got various kinds of proteins, small proteins, medium sized proteins, and even a matrix of complex proteins such as serum.
Okay, so serum. So this would be for a biomedical application. Would that be the case?
Yeah,
Okay. So you have a sponge. It's got trypsin in it. Is trypsin attached to the sponge inside, or is it just floating around inside?
it's attached,
It's
covalently
attached,
attached.
covalently attached to the sponge. It's in there, and I guess the key factor is what you say. It's active for how many days?
For more than 10 days.
More than 10 days, and you can use it 40
Yeah,
times at least. That's a lot. Does the activity reduce over time, or do you have good activity all throughout?
it it reduced, but not that much.
Okay, it reduces a bit, but it doesn't reduce that much. 40 times. That's a lot. I don't know of any enzymes that would act for that long. But trypsin, I guess, is a pretty stable enzyme or a pretty potent enzyme. Okay, I'll have to think about this.
Okay.
So when you want to sorry when you use it to break down enzymes, how long is one time when you say you're using it one time?
We incubate the sponge with the target protein for like about 12-hour minimum.
12 hour, and you
Yeah,
can do this 40
yeah.
times.
We can take it out, put it back 40 times.
40. Okay, that's now making me doubt this statement. Okay, let's listen to your next ones and
Okay,
then yeah.
so I will try to make it more easy now.
Mhm.
One of my hydrogel actuators, soft robots,
Mhm.
was inspired by origami,
Uh-huh.
and it can unfold autonomously
Okay.
inside the stomach
Uh-huh.
for bioapplication or for drug release purposes.
Okay. So it's a soft robot. It's a protein-based material that can unfold on its own.
Mhm.
What makes it unfold?
The triggers that exist in the environment where the robots exist,
Uh-huh.
for example, the stomach,
Uh-huh.
the acidity, the enzymes.
Okay.
Mhm.
So the enzymes and the acidity in the stomach makes this soft robot like protein material unfold.
Mhm.
And then what does it do in there in the stomach?
It can catch the mucin coating layer
Uh-huh.
inside the stomach,
Okay.
so it can increase the retention time of this soft robot inside the stomach
Uh-huh.
to be able to increase the availability of the drug, of
Yeah, okay.
the drug release inside the
Okay.
target site inside the stomach.
Okay. And this was inspired by origami, you
Yeah.
say. What about origami kind of inspired? Is there a folding that's actually happening
Yeah.
the material level, or is
Yes.
it more at the protein level?
Both.
Okay. Okay. So the pH changes, the material folds and unfolds, and you have somehow. Okay, how have you structured the material? How do you fabricate them?
We can use molds. We can
Uh huh.
use 3D printing.
Okay. 3D printing and molds. So to make this kind of origami folded structure, do you use 3D printing or molds rather? What works better?
3D printing.
3D printing. Okay. 3D printing. And if you say origami, I immediately start thinking of the origami crane. Is it a very complex structure that it folds into
It's
like
way
that?
more simple than origami, I think.
Okay.
The paper one,
Yeah.
the paper origami. It's
Okay.
a little bit more. Yeah, it's more simple, I think.
It's more simple.
Yeah,
Okay. But it's
it's way more
inspired
simple.
by
Yeah.
origami. Okay. So the statement is basically that, yeah, this goes, it can go into your body, it can fold or unfold in a certain way, and then stick inside your gut and deliver drugs.
Yeah.
For how long? Do you have a feeling?
Maybe for one cycle of food,
Okay.
like digesting one digesting cycle.
Because the pH changes or
I
so, or that
think
the
so.
enzymes disappear?
I
Okay.
think so. Yeah.
Okay.
Didn't think about the enzyme disappear, but maybe yeah.
Yeah, okay. Interesting. Okay. This, yeah, this sounds like it could be something that is true. But okay, let's go on to your third statement.
Okay. My third statement.
Mm-hmm.
Uh, we developed a system
Mm-hmm.
that we called it APMIS for
Mm-hmm.
synthesizing functional hydrogel microparticles
Mm-hmm.
or protein-based microgels.
Mm-hmm.
And actually, this system was inspired by the process of brewing espresso.
Okay. So the process of brewing espresso. What is APMES?
APMIS is aided porous medium emulsification system.
Aided porous medium emulsification system, and I'm trying to see if how that connects with espresso.
Yeah.
Um, what in the espresso preparation is related to this APMES?
What happens in the espresso, you can have emulsification inside
Uh huh.
the grinded coffee capsule,
Uh huh.
where water flows into these grinded particles between the grinded particles,
Uh huh.
and it collects the oil of the coffee. And inside this capsule or inside this porous medium of the grinded coffee,
Mm-hmm.
the both phases of water and oil mix, making the emulsion, making droplets of oil, coffee oil
Uh huh.
inside the whole phase of water. So
Okay.
these micro droplets that you happens there between due to the mixing of water and coffee oils
Uh-huh.
makes this brewing process.
Okay. Wait, now I'm getting a little bit better. Yeah. So you have in your system what you said, the APMES system. Um, then you're going to use it to make microparticles or
Hmm.
microgels,
Hmm.
I guess what you said. Um, and the APMES system basically uses this espresso idea and mixes oil and water into an emulsion. Is
Hmm.
that the case?
Hmm.
Okay. Okay. Uh-huh. What is it that allows it to mix? Because I know quite a few different ways in which you can mix oil. I mean, you can just take oil and water, mix it really, just by hand or vortexing it in,
Hmm.
and have a detergent in there, and then you have an emulsion.
Hmm.
And it's used a lot in food. Different
Hmm.
ways of preparing these kind of things. Um, so what is special in this espresso method that you were able
What
to
is this?
adapt?
What is special and unique about this method that we are able to make functional protein microgels in seconds instead of waiting for minutes or hours?
Okay.
And then it's very a cheap system that can
Uh-huh.
be scalable, as I said, cost effective,
Yeah.
very simple to handle and work with,
Okay.
and it's the production rate of this system is very high.
Okay, can I ask? Maybe this goes a bit deeper into it, but if it doesn't give away the truth or the lie, can I ask, what is it that actually forces these oil and water to mix and form droplets? So is it a vortexing thing, or is it like a microfluidic thing, or
You
I don't know.
to what makes them mix inside the
What makes them mix and then form these droplets? Are those,
it's
yeah?
because water and oil doesn't mix
Mm-hmm.
at all. So whenever they meet each other in the porous medium,
Are they in a porous medium? Sorry,
yeah,
I think I missed that. Yeah.
the two phases meet in the porous structure,
Uh huh.
and there the emulsion starts to happen.
Okay.
Exactly as the water goes into the coffee grind
Okay.
medium, and where you start to have your coffee in the pot.
Okay. Well, I have to confess I don't drink coffee, so I'm very illiterate about coffee
Okay.
related processes. But of course, I've seen coffee machines
Yeah.
and so on. Okay, porous medium. I think I missed that when I was thinking of the other stuff. When you say porous medium, is this connecting to your first statement about sponges? Do you use those sponges, or how does that work?
Actually, the porous and the sponges are similar a bit.
Okay.
Yeah, I didn't think about this before, but
Okay. So, well, then you don't use
now,
your sponges to make these.
yeah.
Okay. That's
Okay, that's a good thing. Now I'm telling you what is okay.
so okay. Can you say what actually you use as the porous medium? Like, what is
We
this?
use polyethylene
Okay.
frets, FRITS.
Frits. Okay,
Yeah,
so the things that we have in columns at the bottom. I mean,
exactly.
yeah.
Yeah,
Okay.
exactly.
And those are porous. What sizes are these pores approximately?
Wow,
Like,
it depends on the making of the frets itself, on the
okay.
particles that were centered inside the fret.
Uh huh.
I don't remember, but it's in micro-sized.
Micron sized.
Yeah,
Okay.
micro-sized tunnels or pores.
Okay.
Yeah.
Okay, micron size. And so then the microgels that you get, are they also micron size?
Yes.
Like, I mean,
In micro, yeah,
really
yeah.
small microns, or is it like hundreds?
Five to two to five to up to now 40. We can,
5 to 40 microns. So it's
yeah,
pretty small. And is
yeah.
it monodispersed? Like, is there a
It dep
wide range of
it
distributions?
depends on how fast you pass the two phases through the porous medium,
Okay.
and it depends on the tortuosity of the porous medium. It
Okay.
depends also on
Well, sorry, wait one second. Maybe
the
what is a
tortuosity.
tortuosity? I've
It's
not heard
like
this word
the
before.
how complex is the structure, microstructure of the fret. How
Ah,
what is the path length that the particle needs to pass through to
okay.
be to pass from one side to another. If
Huh.
the tortuosity is too high, takes more time. The particle will
Ah,
be more like
retained
yeah,
inside. Yeah.
yeah,
Okay.
and be exposed to more shear stress as well.
Ah. Okay.
So
Okay.
that affects the mechanics of the particle formation and the particle size at the end.
Okay. So this higher tortuosity gives you smaller particles, or
Yes,
is it correlated that way?
yeah,
Yeah.
yeah,
Okay.
you can see that.
Okay. Ooh. Okay. Okay. So I think I've dug in quite deep into this one, but it's
Yeah,
very fascinating.
yeah, thank
I
you.
really like it. Maybe one last question. So you said it's like espresso.
Mhm.
Is it also like something that you push?
You
Like
can,
is
you
it a
can,
pressure based thing?
yeah,
Okay.
you can push it by pressure. Yes, of course.
Okay. So it's very close to this coffee machine
Yeah,
idea.
yeah. We do it also. Maybe we can maybe do it with centrifuge, but
You can do it with centrifuge,
yeah.
okay, but you can also press and actually, I mean, I have heard a lot about different kind of microparticle, microgel. There's a lot of microfluidics related
Yeah,
work on
of
making
course.
these kind of micro droplets and nano droplets and so on.
You're right.
And I'm wondering, like those technologies are quite complex, right? Quite
Hmm.
complex, relatively expensive. Um, and yeah, you make very kind of same size droplets very
Yeah.
reproducibly, and what you're saying is that it's much faster, and if it's just a fret and like pushing like oil and water through it and so on, that sounds like much cheaper.
Hmm.
And then you're saying you get pretty good size distribution
Yeah, pretty good.
of
Not
your
not like
microgels.
microfluidic,
Uh-huh.
but way more fast and cheap and effective than
Okay.
microfluidic.
Okay. Interesting. It's difficult for me to judge if this is a truth or a lie because on the one hand I also know you can just mix oil and water in a certain way and you get some kind of droplets. I mean, you don't have much control over the
All
process
right. Yeah.
there, and on the other hand you have microfluidics that give you complete control but are super expensive and so on. And
Right.
I'm really struggling to wonder if just having a porous medium and pushing oil and water through it actually does the job. Uh,
We will know.
yeah, okay, okay.
Uh,
So let's move on to your fourth statement and
yeah. The
then
fourth
yeah.
statement is I once printed a protein-based material
Uh-huh.
that I tested it in the International Space Station. The
Okay.
aim was to, in the future, to take it to the outer space
Uh-huh.
and see how the zero gravity conditions affect these materials.
So it's been tested in the International Space Station right
That's
now?
we want to,
Okay.
yeah, and the aim is to test it in in zero gravity
Okay.
conditions out of in the space.
But it's not been to space yet.
Not yet, but
Okay.
that's the dream. We have the material. We know how
Okay.
it will behave, but
Why what advantage does it provide in space? Why do we
the
need?
zero gravity conditions,
Mm-hmm.
the stiffness of the material, how it will change, the swelling
Uh-huh.
behavior, the actuating.
Also, this is one of your soft robot
Yeah,
materials?
yeah.
Okay.
So we
Uh-huh.
want to see how all these conditions affect this behavior of actuating behavior.
Okay. In zero gravity. Would it have a specific application in space?
First of all, let's see what happens with it
Okay.
compared to what happens in the lab here on Earth.
Okay. Uh-huh.
That would be interesting.
Do you already have a contact to take it out to space, or how does
We
that
can
work?
we are able to mimic the situation, the conditions
Are
for
you
now.
mimicking it in the lab, the zero
Not in the lab,
gravity?
but in the institute where I work in the
Yeah, okay.
yeah,
In the institute, you're able to mimic
yeah,
zero gravity conditions
yeah.
on Earth. How does that work?
We just use centrifugation based system
Okay,
to make the zero gravity or
okay,
I'm not familiar with what they are doing
uh
there.
huh.
I just give the material and they
Okay.
do it by
So then there
themselves.
is a way to actually mimic
Yeah,
the zero gravity
yeah.
condition, and you see what happens. Are you able to see the material when it's under
They
this
film it for
condition?
us.
They film it.
Yeah,
Okay.
they film it.
Ah.
We don't experience the same conditions that the gel
Okay.
like is experiencing of no gravity thing, no gravity forces.
Yeah, yeah.
Yeah,
Do you already know what happens in these kind of zero gravity conditions? Like
I
what
know that we can like
does it still fold the way?
the we're still working on this,
Uh-huh.
but I think it will fold better than with when there is a
When
gravity.
there is gravity.
Yeah,
Okay. Okay, that's nice.
that's what I think.
Yeah, interesting. So it's a material you have printed already. You have
Yeah,
tested under
3D
zero
printed
gravity
using our 3D
conditions.
printer in the lab, and
Yeah.
that's what we,
Okay, and
yeah,
you want to send it to space.
yeah.
Is there already a plan to have it in space? Like, is there a
Still
timeline?
not, but we
Okay.
are still checking how or investigating how it behaves in
Okay.
similar condition in labs.
Okay. Okay.
Yeah.
Cool. Yeah. Okay. So that's the fourth statement. Um, Let me recap through your other statements from the start. So the first statement is that you have a protein-based sponge,
Mm-hmm.
right? It has trypsin, and the trypsin is active for 40 days, no, for 40 cycles for more than 10 days,
Mm-hmm.
which seems like a lot. Okay, the next one is an origami-based folding material um, that can go into your that can go into your stomach and release drugs. It can
Mm-hmm.
attach to your stomach and then release drugs.
Mm-hmm.
The third one is your what you call your espresso appmes
Mm-hmm.
system, in which you can make microgels. And the fourth one is a 3D printed material that you want to send to space,
Right.
and you've tested it under zero gravity
Mm-hmm.
conditions. Okay, um, I'm I'm kind of doubtful about statement one and statement four actually. One, because I mean, I do work quite a bit with enzymes. It's very difficult to believe this 40-day thing.
10 days.
Sorry, 10 days, 40 cycles thing. Um, and number four, which is the zero gravity stuff. I mean, it could be true. Although it was the one where you gave me the least amount of details, I feel. But I also didn't probe it too much. Uh, I want to say statement four. Is that the lie?
Right.
Ah, okay.
Yeah. Unfortunately,
Oh, yeah.
none of my materials have reached space yet.
Okay.
But if NASA is interested,
Okay. So NASA, if you're listening,
yeah,
yeah,
I will give them some samples.
awesome.
Maybe
Yeah.
they can try them there, see how it works.
Really cool. Yeah. But I'm going to get back to your first statement because I
Yeah.
was almost going to go for that as the lie
Yeah.
because it didn't sound possible. Right? Like what is it that's now you have the sponge. What is the sponge made of? How is the trypsin attached to it and why is it active for so long?
The sponge is made out of proteins,
Uh-huh.
especially albumin and trypsin,
Uh-huh.
and we mix them with tween 20, which is a surfactant,
Uh-huh.
a very biocompatible surfactant to proteins, so it will not affect the structure of the proteins.
Uh-huh.
And we use also photo cross linkers such as APS, ammonium persulfate, and the ruthenium.
Uh-huh.
And whenever we mix all these compounds together and we use a homogenizer, we make a foam. Then we take the foam, we expose it to white light.
Mm-hmm.
So we have a photo covalent, photo cross-linking
Okay. Yeah.
reaction
Uh-huh.
where we cross-link between the tyrosine amino acids that exist on the surface of the proteins that is inside the hydrogen precursor.
Okay.
So trypsin will be covalently attached to BSA,
Uh-huh.
and BSA will be attached also to other BSAs and trypsins.
Okay.
And the foaming process will make this spongy structure due to the air that is entrapped in the solution during the foaming.
Okay.
So we make the foam, we
Yeah.
make the porous structure of the foam,
Yeah.
then we expose the whole foam to white light. So we cross-link covalently all the compounds, all the
Uh-huh.
proteins together. At the end, we have this sponge.
Okay.
So the trypsin is covalently embedded inside the sponge,
Okay.
which increase its stability
Uh-huh.
and
Uh-huh.
activity.
It increases.
It's not
Okay.
active as a free trypsin because some
Uh-huh.
of the active sites of trypsin will be not accessible to proteins due to
Yeah.
their random mixing,
Uh-huh.
but we can keep it working for longer time than free trypsin in a solution.
Okay.
So it's a trade-off.
Yeah, okay. Okay, interesting. So the more you talked about foam, the more I was thinking about your espresso point. And is this also coffee inspired? Because you have this.
No, no,
No, okay,
actually no, no,
okay,
actually
yeah,
no. It was just the aim or the vision was how can we make a spongy or highly porous
Uh-huh.
protein-based material.
How is the foam made then, actually? Like, how do you get the air in?
We just mix with a homogenizer.
Ah, okay. And
Like
then
as you mix, as you
it's
make
like in baking,
exactly,
how you get
yeah,
the air.
exactly,
Okay, okay,
exactly.
okay.
So you as you make the meringue,
Yeah, yeah,
yeah,
okay.
you just
So yeah, you also said albumin, which is, very
exactly,
much like the egg's albumin, I
exactly,
suppose. Yeah. Is it the
exactly.
same albumin or is
No,
it
but
no?
it's
Okay.
very similar.
It's very similar.
Yeah,
Okay.
yeah, but exactly, yes.
Okay.
It's the you form,
Yeah.
but the protein can form by itself, but also
Uh-huh.
we help it with the tween, the
Okay.
surfactant, to make this kind of air entrapment bubbles
Okay.
inside the material.
Interesting. Yeah. So your third statement was espresso food related. This first statement also has a food related connection, which is quite
Right,
interesting to
right,
see.
right.
Um, and but getting back to the point a little bit, so your trypsin is trapped in there, and that is what stabilizes it and keeps it active for that long. Is
Yes,
that the case?
exactly,
Okay.
exactly. It's not fully active, I have to say,
Uh
as
huh.
free trypsin.
Yeah.
It's not
Sure.
accessible
Okay.
as
Yeah.
free trypsin, but it stays there for a longer time.
That's
So whenever you
yeah.
immerse the sponge, you
Yeah.
can take it out, wash it,
Uh huh.
and use it again. With
Okay.
free trypsin, you cannot do it.
Yeah. Yeah. Yeah.
Yeah, that's the
Absolutely.
only.
So what you said you could use it for serum and so on. What is the actual application you're going for with this?
We can use it for like digesting for catalysis, for like digesting oils, for example,
Ah, okay.
for lipids,
Yeah.
for many other applications.
Okay.
It's a concept.
Yeah,
We
okay.
start with something,
Okay.
and after that we learn the system as material engineers.
Yeah, okay.
We learn the limitations. We learn the capabilities of our platform,
Yeah.
and then we start to take it one step left, one step right,
Yeah, okay.
to see what can we do it with,
So
what can
basically
we
you
do
can
with it
have
next.
other enzymes inside.
Yes,
If you're talking about
that's
lipids, if you're talking about oils and so on.
yeah,
Yeah.
that's the plan.
Yeah,
Yeah,
okay.
but every what is nice about proteins
Mm-hmm.
or enzymes, which is a subclass of protein, that each one of them behaves differently,
Yeah.
and that's
Yeah.
one of the
That's
challenges. But yeah,
that's what keeps life exciting.
yeah, exactly.
That's true. Okay, so yeah, so the first statement was that, and that's really fascinating that it keeps trypsin active for so long, and that you have this much activity still after repeated uses.
Mhm.
How big are your sponges actually?
We can mold them in any
Any
size.
size?
Yeah.
Is there a limitation in terms of the foaming or so that?
Um, I think so. Whenever you start to make them like this
Uh
size,
huh.
you need different tools.
Okay.
But we can reach like almost like this size.
Okay. Okay. That's yeah.
Yeah.
So a few
Yeah.
centimeters. Big. Yeah.
You also want to make it go inside the falcon that exists in
Yeah.
your.
Sure.
Yeah.
Yeah.
You cannot make it too big and you cannot make it too small.
True. True. True.
So
Yeah.
yeah.
Okay, interesting. Um, then the next one that you talked about was your soft robot, right?
Mhm.
Like the soft robot made of proteins. And you said that there are these triggers like pH and so on that make it fold.
Mhm.
What is it in the pH that actually makes the whole thing fold?
Well, let me start with the first thing about the most important thing about proteins.
Uh huh.
As you also know that proteins are existing in their folded state
Mm hmm.
in physiological conditions in their normal state.
Mm hmm.
When you expose a protein for like different triggers
Mm hmm.
or conditions such as acidity,
Mm hmm.
such as light,
Mm hmm.
such as enzymes, salts,
Mhm.
high salt, a solution with high salt
Mm hmm.
high concentration of salts, the proteins their structure can be interrupted or
Okay.
disrupted.
Disrupted, yeah.
Yeah,
Uh huh.
and they can deform.
Deform,
Yeah,
okay. Uh huh.
or denature. Like
Yeah.
yeah,
They unfold.
yeah, they totally
Okay.
unfold.
Uh huh.
But what was nice about proteins that thermodynamically,
Uh huh.
whenever you just release them from this trigger,
Yeah.
they you know how to remember and go
Okay.
to their initial state
So
of
they remember their original shape.
exactly
And just from the basics, basically, these proteins are chains of amino acids.
right.
They remember their original shape when
Yes,
they're in the right conditions that's
exactly,
suitable for them. And if
exactly,
you change the conditions, now they unfold.
exactly,
And this translates into like a macro scale folding in your
exactly,
material.
exactly,
Interesting.
exactly.
Is it similar, like the soft robotics kind of thing that you said? Is it a similar kind of structure as what you explained with the sponges, that it's attached through a polymer and so on?
Actually, yes, we
Mm-hmm.
incorporate or we embed the proteins
Mm-hmm.
inside a passive hydrogel matrix.
Okay.
We can use different polymers.
Uh huh.
These days, we are focusing on PEGDA,
Yeah.
polyethylene glycol deacrylate,
Uh huh.
and we can do that with any protein matrix.
Okay.
But some of the proteins, as I mentioned, they are passive. They don't react or they don't respond to triggers in the environment.
Yeah.
But proteins has this nature ability
Uh huh.
inherently. They inherently has this smartness.
Yeah.
So that's how we combine these two worlds
Interesting.
in order
Very fascinating.
to control the exactly as you described it, to control the actuating behavior
Uh huh.
through the actuating behavior of the protein itself.
Wow. So something happens at the nanoscale and you see it translate into the macro scale
Exactly.
by what you
It
can see by
happened.
eye.
The scientists developed this before with polymers, like
Yeah. Mm-hmm.
thermoresponsive polymers, many polymers. But
Mm-hmm.
you need to design the
Mm-hmm.
polymer to have this property.
Yeah.
With proteins, you don't even.
They're already designed.
Yeah,
Yeah.
exactly.
Plus, you can design them more, I
Ex-
suppose, nowadays. Like you can tune their design with a lot of these
exactly.
AI tools and so on
Yeah,
that have come out.
right. I agree.
Interesting. Yeah, a lot of potential then with the developments that are coming up for
Mm-hmm.
sure. Yeah. Cool. So so there's all of these examples are with proteins, which kind of brings me to the segment of what is your favorite material? Is it something protein?
Uh, our favorite material is for sure proteins,
Uh huh.
especially what we are working on these days a lot
Yeah.
is a cheap protein that is really abundant in our
Uh huh.
plasma, which is BSA. Or
Okay.
in our plasma, it's a human serum albumin,
Yeah.
but we mostly use bovine serum albumin.
Uh huh.
They are very similar, but there are some amino acids that are different in both proteins.
Uh huh.
But this is our main protein in the lab.
Okay. So for just
We
for our
s-
listeners,
we try with it everything. We
wonderful.
start with it every application,
Yeah.
and after we see how it works, we move toward more complex, more expensive, more
Yeah. Okay.
low abundant or rare proteins that we can have.
Can you just for our listeners, can you just describe what you're holding there?
This is BSA in its powder state
Mm-hmm.
crystals.
In a glass vial.
Yeah, and
Yeah.
it's now it's in a glass vial. It's not that harmful, to be honest.
Mm-hmm. Is it protein, like how, if I talk to someone at the gym and they say, eat a lot of protein,
Yeah, yeah.
it will work the
This
same? Yeah.
exists in the blood
Yeah.
of cows, you know.
Okay. So
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 if
it's
you eat
really good healthy stuff.
yeah, if you eat
Yeah.
a steak,
Yeah.
buy steak, you get some proteins,
Ah. Okay.
albumins, other proteins, you know.
And is that why it's also so cost effective for you to use? Like because
Yes,
it's very abundant.
yes. BSA
Okay.
is that's what is unique about it. It's very
Mm-hmm.
stable.
Uh huh.
It has a diverse biochemical structure.
Yeah.
It can interact with many molecules inside the body,
Uh huh.
like small molecules, protein fragments. It's a carrier,
Uh huh.
and it has a long half time life. What
Half life. Yeah.
half life time for of
Yeah.
about, if I'm not 21 days.
Okay.
So it's
In the
pretty,
body.
yeah. So
Yeah.
it's very stable.
Interesting. Yeah.
So it can
Nice.
interact with many other. It can be functionalized.
Yeah.
It's enough big. Like it's about 67 kilodaltons.
Okay.
So you can use the diverse structure of the surface of BSA to
Okay.
be able to functionalize it with different polymers.
Wow.
So
Okay.
that's
Yeah.
why we use it. We start with it. We start every experiment or every material with
Uh-huh.
BSA.
Nice. Yeah. So it's a relatively cheap protein.
Yeah.
It's stable. It actually ticks a lot of boxes. It's stable within the body. It's yeah, able to you're able to bind other proteins to
Yes.
it, and then it has this folding, unfolding property that
Exactly.
you
That's what
really
I
need.
forgot. Thank you.
Yeah.
Thank you. It's a globular structure.
It's a globular structure.
Yeah,
Okay. Uh
it's
huh.
like a sphere structure protein,
Okay.
which can, as you describe it, it can be unfolded or refolded.
Yeah.
Yeah.
Okay.
Yeah.
Nice, wonderful. Because I know BSA quite a lot. Also, like anytime anyone who works in biology uses BSA as just this background protein for you put it in cell culture, you put it in for protein purification. You have it also in materials in biomaterial science. You
Right.
have it in the background to prevent things from sticking to other things
Yeah.
in the body. So it's such a commonly used protein,
Yeah.
but you make such advanced materials
Yeah.
out of it that can form sponges and soft robots and
That's
so on.
right. Yeah.
So that actually brings me to actually the final part of this. You're making these really cool protein-based materials that can change shape, that can be active as sponges,
Mm.
that can form microgels and so on, and maybe one day go into space. So if you think of whatever research you're doing, everything works out, it's working beautifully, and 20 years in the future you got protein materials out in the world. What would you envision the world being like at that point? Like, what would these materials be out there doing?
I think where the future is heading is toward materials where are not any longer passive.
Mm-hmm.
They will be very interactive materials.
Mm-hmm.
They will be these materials will be responsive,
Mm-hmm.
and I think with synthetic biology and all the AI tools and all these protein engineering
Mm-hmm.
progress that we have seen recently, also in the Nobel Prize thing,
Yeah.
I think the materials can have the decision making ability.
Oh.
So they know when to interact, with what to interact, and where to interact.
Okay.
They can decide. Proteins can be very smart
Mm-hmm.
with time and all these AI tools that
Uh-huh.
they decide if it's the right time to interact with molecules or just be passive. Now it's not the
Okay.
time to do the function that I'm designed to do it.
Okay.
So that's what I think where the smartness of proteins together with the smartness of the science that we are
Mm-hmm.
trying to improve these days,
Yeah.
I think it will go to materials that they can just decide by themselves.
Wow, that's fascinating. So it's not just computers with AI and so on that getting smarter and smarter, but our materials are also going to be incredibly
Yeah,
smart
so
in the future.
some of the materials that exist today are really smart.
Uh-huh.
They just respond to specific triggers in a specific environment. For example, for drug release in cancer
Mm-hmm.
tumors, that
Yeah.
this is very they stay passive until they reach the site of
Mm-hmm.
the tumor. Then they start to be active.
Mm.
This kind of decision making material,
Yeah. Yeah.
but we can improve it way more just to release it and forget about it,
Uh-huh.
and it
And
can
then
just yeah
it can do multiple
yeah
functions
yeah
and do know when to do those
yeah
functions.
exactly.
Yeah, that's going to be very exciting to see in the future.
Yeah, and also I'm thinking one thing that is really the future is going, where I see the future is really advancing with protein materials or with
Hmm.
material science in general, is to make the materials inside the body.
Make the material inside
Yeah,
the body.
like
Uh-huh.
if already now there's a kind of uh, amazing achievements of printing,
Yeah.
3D printing inside the body.
Oh, wow.
So
Okay.
you don't need to print
Ah,
the
you mean in a surgery sort of
yes
setting and
yes
so on.
in
Yeah,
situ
okay.
on demand,
Ah,
to have a
okay.
system that you just print or form the material
Okay.
inside your stomach or inside
Yeah.
on your heart or anywhere
Yeah.
in your body without any like preparations before, or in
Okay.
the lab or any pre processes that
Mm-hmm.
you need to do, and you can save time,
Yeah.
yeah, shorten the surgery
Mm-hmm.
time. I think this is what is really going to be interesting in the next 10 years, to be able to form it in situ.
Wow. And coming back to the point where you said they would be smart and they would be able to make decisions, do you think that these materials that you print in the body would also be smart enough somehow to know, okay, this is where it's going to form a certain tissue,
Mm.
at a certain time it's going to change its mechanical property,
Exactly.
to fit certain
I think
regenerative
that's
processes and so on?
every material scientist dream.
Yeah. Okay.
And if we and I think the material science in
Mm-hmm.
general is the key for many problems that we will face in the future,
Yeah.
especially in agriculture
Mm-hmm.
with
Mm.
water.
Yeah.
So we
Okay.
can they already design gels that can use to for plants to grow
Mm-hmm.
plants and trees and many things. So I think if we can make them more smart,
Mm-hmm.
that they can be adaptable to many environmental conditions, that's something that will help the to improve the future of of you know of every application.
Wonderful. Yeah.
Yeah,
That sounds really interesting. So the future is a future of protein-based biointelligent materials that are smart enough to enhance all sorts of applications, biomedical, agro.
yeah, but it depends also. It's not possible without all the tools that synthetic biology AI are introducing to science these days. We need to make the proteins more scalable,
Mm-hmm.
highly with high production rates,
Yeah.
and you know that better than me. So,
Yeah, that is a
so
challenge, but
yeah,
of course, this question is mainly about having a fantasy of the future, imagining the future as a fantasy. If what your research, what you're developing in your research actually all works out
yeah,
and it's technologically scalable,
yeah,
then yeah,
yeah.
I
One
think.
and also one curious question, like or make me curious about it is when we made polymers from
Mm-hmm.
molecules. Now we can make materials from proteins.
Yeah,
What is the next materials? What are the next molecules that can we make out of them? Like this, I mean biomolecules.
yeah,
Is there
interesting.
something different with? I don't know.
There's polysaccharides, there's different lipids,
Yeah,
there's different
yeah.
various
What
different
can
types
we
of
invent? What we can introduce
biomolecules.
more?
Yeah, there's
Yeah,
it's a fascinating world that we
yeah,
have
yeah.
ahead exploring, and I think I learned a lot in this conversation. I think it was very fascinating. I think your statements were very well crafted. I'm happy I was able to find the lie, and I'm happy that your other statements were very true. It was very interesting to learn about that. So thanks a lot.
Thank you so much for having me today.
Yeah.
It's a real pleasure. I
What
enjoyed it a lot. Very nice experience.
Wonderful. I'm glad you enjoyed it. I'm sure our viewers and listeners have also enjoyed this entire conversation. So yeah, we're on YouTube, we're on Spotify, we're on Apple, we're everywhere. So yeah, find us, follow us, like, subscribe, all of that, and look forward to the next one. See you.
See you. Bye.